Hello, welcome to Biology, the scientific study of life. Morphological and molecular homologies. In addition to fossil organisms, certain morphological and molecular similarities among living organisms can indicate phylogenetic history. Recall that similarities due to shared ancestry are called homologies. For example, the similarity in the number and arrangement of bones in the forelimbs of mammals is due to their descent from a common ancestor. With the same bone structure. This is an example of a morphological homology. Notice this figure. In the same way, genes or other DNA sequences are homologous if the nature of their similarity suggests that they are descended from the sequences carried by a common ancestor. In general, organisms that share very different or very similar morphologies or similar DNA sequences are likely to be more closely related than organisms with vastly different structure or sequences. In some cases, however, the morphological divergence between related species can be great and their genetic divergence small, or vice versa. For example, consider the Hawaiian silversword plants. These species vary dramatically in appearance throughout the islands. Some are tall, twiggy trees, and others are dense, ground-hugging shrubs. Notice this figure, notice that. Boom. But in spite of these striking phenotypic differences, the silversword genes are very similar based on these small molecules divergence. Scientists estimate that the silversword group began to diverge 5 million years ago, which is also about the time when the oldest of the current islands formed. We can infer that the morphological diversity of the silversword is controlled by relatively few genetic differences. Sorting homology from analogy. A potential red herring in constructing a phylogeny is similarity due to convergent evolution, called analogy, rather than to shared ancestral homology. Convergent evolution occurs when similar environmental pressures and natural selection produce similar analogous adaptations in organisms from different evolutionary lengths. Notice this figure also. For example, Australian and North American. Burrowing moles are very similar in appearance. However, their reproductive systems are very dissimilar. Australian moles are marsupials. Their young complete their embryonic development in a pouch outside the mother's body, whereas North American moles are eutherians. Their young complete their embryonic development in a uterus within the mother's body. Indeed, genetic comparisons and the fossil cord provide evidence that the most common ancestor lived 120 million years ago, about the time that marsupial and eutherian mammals diverged. This ancestor and most of its descendants were not moth-like, but similar characteristics evolved independently in these two moles' lineages as they adapted to similar lifestyles. Distinguishing between homology and analogy is critical in reconstructing phylogenies. For example, both bats and birds have adaptations that enable them to fly. This is superficial resemblance might imply that bats are more closely related to bears than they are to cats, which cannot fly, but a closer examinations reveals that the complex structure of the bat's flight apparatus is far more similar to the forelimbs of cats and other mammals than to birds' wing. Fossil evidence also documents that bat forelimbs and bear wings arose independently from walking forelimbs of different ancestors. Thus, we can state that the bat's forelimbs or forelimb as homologs to those of other mammals, but analogous is in function to a bird's wing. Analogous structures that have evolved independently, such as bat forelimbs and bare wings, are also sometimes called homoplaces, from the Greek for to mold in the same way. Besides searching for incorporating similarities or fossil evidence, another clue to distinguishing between homology and analogy is to consider the complexity of the characters being compared. The more points of resemblance that two complex structures have, the less likely is that they evolved independently. For instance, the skulls of a human and a chimpanzee do not consist of a single bone, but rather of many bones fused together. The compositions of the two skulls match almost perfectly bone for bone. It is highly improbable that such complex structures, matching in so many details, have separate origins. More likely, one gene involved in the development of both skulls were inherited from a common ancestor. The same argument applies to comparing genes themselves, which are sequences of thousands of nucleotides. Each nucleotide position along a stretch of DNA or RNA represents an inherited character in the form of one of the four DNA bases: A adenine, G guanine, C cytosine, or T thymine. Those comparable regions of DNA that are 1000 nucleotides long provide 1000 points of similarity or difference between two species. Systematists compare long stretches of DNA and even entire genomes to assess relationships between species. If genes in two organisms share many portions of their nucleotide sequences, it is highly likely that the genes are homologs. Evaluating molecular homologies. Molecular comparisons of nucleic acid often pose technical challenges. The first step is to align comparable nucleic acid sequences from the two species being studied. If the species are very closely related, the sequences likely differ at only one or few sites. In contrast, comparable nucleic acid sequence in distantly related species usually have different bases at many sites and may even have different lengths. This is because often longer periods of time insertions and deletions accumulate, altering the lengths of the gene sequences. Notice this figure. Suppose, for example, that certain non-coding DNA sequences in a particular gene in two species are very similar, but a deletion mutation has eliminated the first base of the sequence in one of the species. The effect is that the remaining sequence shifts back one notch. A comparison of the two sequences that does not take this deletion into account would overlook what in fact is a very good match. To avoid this problem, systematists use computer programs to analyze comparable DNA segments of differing lengths and realign them appropriately. Such molecular comparisons reveal that a large number of base substitutions and other differences have accumulated between the comparable genes of the Australian and North American moles, indicating that their lengths have diverged greatly since their common ancestor. Thus, we say that the living species are not closely related. In contrast, the high degree of gene sequences similarity among the silversword supports the hypothesis that they are all very closely related in spite of their considerable morphological differences. The fact that molecules have diverged between species does not in itself tell us how long ago their common ancestor lived. Sometimes, as in the case of the moles, the fossil record provides data about when their common ancestor probably lived, but in the case of the silverworts, few fossils have been found for such species. Researchers may be able to compare their molecular divergence with that found in other plant lineages that have more complete fossil records. These values can serve as a sort of molecular yardstick to measure the approximate time span of various degree of divergence. This is how researchers calculated that the silverwort's common ancestor lived approximately 5 million years ago. Just as with morphological characters, it is necessary to distinguish homology from analogy to determine the usefulness of molecular similarities for evolutionary studies. Two sequences that resemble each other at many points along their length are most likely homologous, but in organisms that do not appear to be closely related, the basis that the otherwise very different sequences happen to share may simply to be coincidental matches or molecular homologies. Scientists have developed mathematical tools that can distinguish distant homologies from such coincidental matches in external divergence sequences. For example, such molecular analysis has provided evidence that despite our lack of morphological similarity, we humans do indeed share a distant common ancestor with bacteria. Scientists have so far sequenced more than 20 billion bases worth of nucleic acid data from thousands of species. This enormous collections of data has fed a boom in the study of phylogeny, clarifying many evolutionary relationships such as those between the Australian and North American moles and those between the various silverworts. Thanks for listening.